Oxygen gets reduced to water. Why is it called reduction? Because he receives negative charge. I wish my bathroom scale would work like that. The more sweets I eat, the more I reduce my weight. Wouldn't that be nice? The more electrons you receive, the more negative you become. You're reduced in charge. So when they say a reduction, they mean it's reduced in charge. It's more negative. Okay, we talked about enzymes. Enzymes run reactions, and enzymes have to fit with their substrate. The substrate is the starter product. That's the molecule that the enzyme's acting on. The enzyme binds to the substrate, and the substrate gets converted into a product, sometimes called an end product. So glucose is your substrate. Not just one reaction it's going to go through. It's going to have multiple enzymes. It's going to run through many reactions. Your end products are going to be water, carbon dioxide, ATP, and heat. So the substrate is what you start with. That's what the enzyme binds to. That's what the enzyme is going to run through a reaction. So the enzyme acts on the substrate. And you get a product. Okay? Let's say I give my patient sulfanilamide, an antimicrobial drug. Might even call it an antibiotic, but it's actually an antimetabolite because it's going to interfere with the bacterial cell's metabolism. Bacterial cell can't convert TABA, a nutrient, into folic acid. Interferes with its metabolic reaction. What if I gave my patient sulfanilamide drug, and instead of the enzyme binding with the real substrate TABA, it binds with the sulfanilamide drug instead? What kind of a drug, what kind of action is that? How did I inhibit the enzyme? Remember competitive? Sulfa drug is a competitive enzyme inhibitor. It binds to the active site on the enzyme, so the real substrate can't bind. And that's how it works, by competitive enzyme inhibition. Remember, it mimics the shape of the substrate. So the drug looks to the cell like the real substrate. It mimics the shape of the substrate. That's a competitive enzyme inhibitor because it's going to bind to the enzyme's active site because it's the same shape as the real substrate. That's where that beta-lactam ring comes in. Penicillins, bacitracin, those kind of antibiotics, they have a beta-lactam ring. That beta-lactam ring is the active part of the molecule. That's the part that binds up the active site on the enzyme. It fits the active site on the enzyme. And let's see. What's non-competitive enzyme inhibition going to do? Yes?
It binds to the allosteric site.
Right, it binds to an allosteric site. And the end result is what? It changes the shape of the enzyme. So non-competitive enzyme inhibition simply changes the shape of the enzyme. Binds to a different site, an allosteric site, that changes the shape of the enzyme. But what kind of agents do we use as non-competitive enzyme inhibitors? You're going to wash your hands with one next week. You're going to use Hippoclene surgical scrub next week. And it contains chlorhexidine gluconate. Chlorhexidine is a halogen. Halogens and heavy metals are non-competitive enzyme inhibitors. They're poisons. They're poisons. Poisons damage the enzymes. They bind to the allosteric site and damage the enzyme. So, what about my enzymes? Poison to me too. So what's the problem with your non-competitive enzyme inhibitors? What's the problem?
They damage your cells.
Right, they're toxic to the host. They can be toxic to the host. So if you're going to use a non-competitive enzyme inhibitor on me, I'd appreciate it if you gave me a really low dose. If you use a competitive enzyme inhibitor, like penicillin, what's the problem now? Bacteria develop resistance, right? They can develop resistance to penicillin. Which one's a better magic bullet? Penicillin
Depend-
would be, so competitive enzyme inhibitors are better magic bullets than non-competitive enzyme inhibitors. Non-competitive enzyme inhibitors are heavy metals and halogens like chlorine and iodine. They can be toxic to the host. What happens in glycolysis? What's glycolysis mean?
So the breaking of sugars, so you break the glucose in half to get to pyruvate.
Good, perfect. And what are pyruvates? What kind of a molecule is pyruvate?
3 carbons.
So it's an organic molecule, right? So you split your glucose in half to get your little organic molecules, and those little organic molecules are going to become the substrate for the Krebs cycle. What do you do with the pyruvate? Aerobically, you're running through your Krebs cycle. So Krebs cycle starts with little organic molecule acetyl, and that actually came from your pyruvates. So in your aerobic cellular respiration, you split your glucose in half. Now you have 2 smaller organic molecules to run through your Krebs cycle. Okay. When you do glycolysis, glucose splitting, that's an oxidation. So breakdown reactions are typically oxidations. Now, you do your Krebs cycle, more oxidation, you're still breaking it down. So your entire cellular respiration, even though your oxidative phosphorylation step is a reduction, they're coupled, your catabolism is cellular respiration. Anabolism is building reactions like DNA synthesis or DNA replication and protein synthesis. There you go. These questions are like, oh my God, they're so easy. I can't believe I was so worried. Oh, they're so easy. Like here's one, okay? Fermentation of carbohydrates to alcohol gives off carbon dioxide bubbles that cause bread dough to rise. What microbe performs that type of fermentation? Who makes your bread rise? Yeast, yeah. Saccharomyces yeast, right. That's easy. Alrighty. Okay. When the cells make an ATP, where does a bacterial cell make its ATP? Plasma membrane. Where does a eukaryotic cell make its ATP? Mitochondria. Good. See how easy that was? What are the 3 steps in aerobic cellular respiration?
Glycolysis, Krebs cycle, and then whatever the last one was.
Oxidative phosphorylation. Good. Fermentation. I split my glucose in half. I got 2 pyruvates. I dump my electrons and hydrogens onto it. I end up with alcohol or lactic acid or butyric acid. How many ATP did I get?
2.
Where'd they come from?
Glycolysis.
Good. Perfect.
Got that.
Okay, let's talk about some genetics type stuff, some DNA replication. When I build my DNA molecule, what kind of bonds are holding it together?
Hydrogen.
And?
Covalent.
Good. Got any ionic bonds in there?
No.
No. You don't want your DNA held together by ionic bonds. You want it to have strong, sturdy side rails of covalent bonds. Then you want to have those nice, flexible hydrogen bonds in the middle so that the molecule can open up, new copies be made in the middle, and then they can close back up again, new copies. And each new copy is half original strand and half new copied strand. What type of replication is that called? Good, semi-conservative. Alrighty. The bacterial cell has 1 chromosome. Remember that one for your final. That's an easy question. How many chromosomes does a bacterial cell have? 1. What's it called? What do you call the bacterial chromosome? The nucleoid. The nucleoid, the bacterial chromosome. Okay? What's the genome?
In the bacteria, it's the plasmid and the nucleoli.
Right. The bacterial cell, it's the nucleoid, and the plasmid if it has one. It's all the genes that the cell has. Remember, plasmid has genes just like nucleoid has genes. All the genes of the organism. It's the DNA. The DNA of the organism and all the genes of the organism. Good. And let's talk about transcription and translation. So I think we did DNA replication pretty good. Okay? Protein synthesis. 2 steps. Your book lists all kinds of other steps. Initiation, elongation, termination. Who cares? This is what you remember. DNA, semi-conservative replication, get DNA copy. Because the whole cell is, or the cell is going to divide into 2 cells. That's DNA replication. Protein synthesis, transcription is the first step. The DNA is transcribed into mRNA. Translation. The mRNA is translated into a chain of amino acids, which becomes a protein. So we're back to the central dogma. DNA to RNA to protein. All right. Did I get any ATP by doing that? No. In fact, I used some. I have to use energy. Sometimes it's even in the form of GTP, which is a type of